Hello, it's Professor Cher. Today I want to talk to you about population genetics. Here's a picture of a ground finch made famous by Charles Darwin's Journey to the Galapagos Islands, and later a lot of work by other scientists, including Peter and Rosemary Grant. And you can see, and you might know the story, the beak is a very particular shape, in this case very fat and good for cracking seeds. And what we found, particularly the Grants over a long period of time, is that the population makeup of those beaks can shift even within a generation or two. They can see the beak depth changing. And so this lecture is going to talk about the ideas that help us describe that change and some reasons why those might happen. First of all, you have to have genetic variation in the population. So if all those finches had the same exact beaks because they had the same exact alleles, the versions of the genes, then there would be nowhere to move to. So the first thing that you have to have is actual changes in the DNA sequence, and we'll see a few ways that that can happen in a moment. And in the end, this has to result in some change in phenotype. If there's a mutation or a change in the DNA, but it doesn't result in anything changing outwardly in the organism, that means that it's not going to make a difference. Now, I do want to point out, though, that the phenotype that I mention here doesn't necessarily mean that it has to be, color of fur or height or something, stereotypically external. It can also be something like enzyme function. In the end, there's some ability of that organism that changes, but it can be on a smaller scale. We tend to, when we talk about genetics, think about discrete characters, as in white pea plant versus purple pea plant, or sickle cell allele versus not sickle cell allele. And that's just two either or characters. But really what most traits are these kind of things called quantitative characters, where we have a continuum. You can see height, they're arranged by height. That is a classic quantitative character. Here's a number of different eye colors. That's a quantitative character. And usually those means more than one gene's involved. So we'll mostly talk about these discrete type characters when we're doing these population genetics. But just remember that the whole situation is actually a lot more complicated with more than one gene coding for a specific phenotype. I mentioned that the variation needs to be genetic. This is an example here of a caterpillar that's the same species, but it just phenotypically looks different depending on what food it's on. And for a few generations after that, actually, even if they get moved to food, the caterpillars will look like this, even if they're not on this food source anymore. But it just lasts a few generations. And we know that that's actually due to epigenetic changes, which means changes in basically the coiling and shape of the DNA, which allows access or not access to that DNA to transcribe. In other words, the DNA is modified temporarily in shape in order to regulate gene expression. So that can get passed on some. We have better evidence in other organisms, but it appears to be not a permanent passing on. So epigenetics may influence population genetics to some degree, but if it's not passed on generation after generation after generation, then it's not going to be the same as what we're going to discuss next. If you're unfamiliar with epigenetics from Bio 1 and you want to read a little bit about that, here's a good website that you can go to. There's 5 main sources of genetic variation that I'm going to have you know. And of course, this is in a population. And I will go through each of these in order. Some of these ideas are in the book. Some of these ideas are in this, the Understanding Evolution from Berkeley, and some of the other ideas I've picked up from other resources. So you'll probably have to write an essay of some sort about these concepts. So you want to prepare for that. You guys know what a mutation is already. This is a change in the DNA sequence. And what we get is, a lot of times we think about new alleles, but you can also get new regulatory instructions, and we'll see that in a moment. In the coding regions, so coding means that the mRNA that's transcribed will be translated into a polypeptide and then folded into a protein, or that the RNA that's transcribed is regulatory RNA itself, like microRNA. There are different types of mutations that you can have. This is all material from Bio 1 chapters, so if you need to go brush up on what these things are, those are present in chapters prior to this. But a lot of times we talk about point mutations. So point mutations are small changes in the DNA. So um, if this is your initial DNA, this is just one little bit of it. And when the DNA is being copied, and remember this has to happen during meiosis copying. So any mistakes in mitosis cell cycle, the S phase when it does the DNA replication, that's not going to matter for evolution, natural selection, passing on of genes. So this has to happen in the meiosis cell cycle. So it's going to be in the S phase when the DNA is getting replicated prior to meiosis. And so DNA polymerase sometimes makes mistakes that are not corrected. So for example, when it copied the DNA, accidentally put in a G here instead of the C that it should have. It didn't get corrected. So when the new strand is synthesized, then you had a C here. So now we have, even though it's both CG pair, it's completely different, right? Because it's on different strands. So now we have a G instead of a C. And there are times when maybe the C would be left out by accident. So that would be what we call a deletion, small deletion. Or sometimes there's a small insertion, like two C's are put in a row by accident. And so those all can cause changes. There are various consequences of those mutations. That's what we have here. When you change the DNA, I'll put a star for mutation, the mRNA will always be different. And I'll put a delta for changes. So mRNA is always going to change, and that's your transcription that happens there because it depends on the DNA sequence. The mRNA changing may cause an amino acid change, may cause a change in the protein. And so I say it may cause an amino acid change because remember that redundancy of codons? Where you have multiple codons that can actually result in the same amino acid. If you don't remember, go look at a codon wheel or a codon chart. But if the mRNA actually, the codons still code for the same amino acid, then there's not going to be any difference. So that's what we call a silent mutation. It's also possible that the amino acids do change and one or more amino acids is different. That's a missense mutation. And then a nonsense mutation is really where we get an early stop codon. And so you get what we call a truncated polypeptide, which means shortened. And sometimes those have various functions, but a lot of times it means it can't fold and it's completely non-functional. And remember, why would the protein changing matter? It could change the shape of it if it folds differently, or it could change what I'm going to call its character, meaning is it hydrophobic regions, hydrophilic regions, positive charge regions, negative charge regions, those kinds of things. And when we look at these, most of the time, the consequences of those changes are neutral, where they actually don't make a difference, particularly if the protein doesn't end up being varied in function. Second, most frequently, they're deleterious, meaning that they are damaging or kill the organism, something like that. And sometimes they're beneficial. Remember, mutations are not good or bad. It just depends on A, the result, and B, the environment, right? And so remember, all these are actually environment dependent, beneficial versus deleterious. And what we'll see when we talk about them in genes for proteins, a lot of times we're talking about the stuff genes. So that's example is the mutations in the MC1R gene for those rock pocket mice that you have seen stories about. That's actually in the gene coding for the receptor MC1R. And you can also have mutations potentially in the body plan genes where you have a change in the transcription factor, which is what the body plan genes code for. And that transcription factor would then regulate the turning off and on of the downstream genes differently. And so you get a different kind of timing cascade going for the development of the organism. Not all changes, in fact, most changes are in non-coding regions. For example, in a switch type region. Most of the genome is non-coding, more than 98% is non-coding. So that's why most mutations actually happen there. Sometimes, a lot of times, it doesn't make a consequence, so it's neutral again. But sometimes if you're in an enhancer region, that's what we're going to call the switch. That's where transcription factors bind to turn on genes and other regulatory molecules to turn off genes. So what can happen here, like with the finch story with the beaks, is that you have a change in the switch, and so that changes the timing and intensity of expression, which results in a different beak shape. You can also actually have mutations at splice sites. So if you recall, when you look at DNA, and then I'll look at the mRNA part because that's when this actually occurs. So in the mRNA, let's see, there's parts that are called exons, E3, and there's parts that are called introns. So it's going to be I1, I2. And typically the introns are spliced out, meaning that they're cut out and you're left with just the exons in your mRNA. And those are what get translated into polypeptide and folded into a protein. And how does it know where to splice? There's actually specific sequences at the junctions between the exons and introns, because I mean, they're all just mRNA and just DNA prior to that. It's not like there's anything special about them structurally. It's just code there that the spliceosome recognizes to bind and to cut. So if there's actually a mutation in the DNA at the spot that's going to be a splice site, then you might actually not splice anymore. And so what would happen would be that you would have the intron actually stay in, and all of a sudden you have a completely different mRNA sequence because you have this whole section here now that's new mRNA that is now going to be translated into polypeptides and make a protein with potentially different function. We can also look at alteration in gene position or number. These are a little bit larger scale. So you'll see that there's a number of things that can happen to the chromosomes, but this is not like gene or D. This is actually a big chunk of the chromosome that gets deleted, or a chunk that gets duplicated, or inverted, meaning that the order of the bases ends up flipping. And there's also translocations, which is where bits of chromosomes that are not homologous, so like chromosome number 1 and chromosome number 10, accidentally switch information. And so now all of a sudden you have completely different genes next to each other. And sometimes that changes what regulatory sequence is controlling the genes next to it now, and that could change phenotypes. We also have, usually those are deleterious or neutral. Here's an interesting example of these fusions of chromosomes and switching up of chromosomes. So like if we look at the mouse chromosomes and we look at sequences, we can actually see those homologous sequences all in the same chromosome in humans, number 16. So obviously they got shuffled around through evolutionary time and ended up in the human lineage on that same chromosome. We also see, so 23 pairs of chromosomes is what humans have. Chimpanzees have 24 pairs of chromosomes. And actually what they see is 2 chromosomes in the chimp, number 12 and number 13, look basically like they're fused to make human chromosome number 2. If you look at the sequences, typically telomere type sequences, the orange parts here, only on the tips of the chromosomes, that's what the telomeres are. But in the human chromosome 2, you actually see telomere sequences right in the middle where that fusion would have happened between those 2 chromosomes. And of course that puts different genes together in different ways with the different regulation and so forth. So these kinds of things that we looked at here are mostly gene position, but what about gene number? So here's a classic example of duplications of genes and then divergence. So the globin gene we can trace that actually. So this is in humans here. We have a ton of genes that are different globin genes. There's actually 2 whole families of them. A lot of them are non-functional now. That's what these green chunks are, but the other ones are slightly diverged from each other. So the idea is that you have an ancestral globin gene, just one of them in a common ancestor of the past. It got duplicated, And then one of those could diverge in one way and the other can diverge in another way, meaning basically they're accumulating different mutations. And actually, this is a very common thing you see because sometimes if the mutation happens in the one gene and that's all you have and is deleterious, then the organism will die. But if you have a second copy of that gene, this one can still go on and do its normal function. That second copy, though, accumulating mutations may not kill the organism because you're not getting rid of the functionality the initial gene had. And so this is a common thing that you see about diverging. And we'll actually look at an example here showing olfactory receptor duplications and divergence.
Actually, when you get down to it, what mostly characterizes a vertebrate genome is just a tremendous amount of gene duplication followed by slight divergence of the genes. We actually have vast gene families where a particular gene has given rise to 2 genes, 4 genes, 20 genes, and they've diverged slightly to take up different functions. It's not very creative. You wouldn't get a lot of points for innovation for this, but it seems to have worked. It's given rise to zillions of immunoglobulin genes involved in immune systems, or intermediate filament proteins involved in epithelial surfaces. You own 111 keratin genes, as best we can count. Growth factors. You own, we think, about 42 transforming growth factor beta genes, whereas flies and worms get by with just 2 related genes of that sort. You have 42 such genes. And the most extreme case of all, smell receptors. You may not realize, but out of the 30,000 or so genes in your genome, 1,000 of them are smell receptors. You have 1,000 smell receptors in your genome, indicating that somewhere in your evolutionary past, some ancestor was very interested in smell as an important modality. In fact, all mammals have about 1,000 smell receptors because that was the principal sense. Now, disappointingly, I've got to tell you that your more recent hominid ancestors have lost interest in this sense. Most of your smell receptor genes, 2/3 of them, are actually broken and non-functional. It's not true in most species, but hominids have lost lots of their smell receptor genes, probably because we got much more interested in sight, but nocturnal animals and other animals have kept their smell receptor genes in better working order. So in any case, this is a lot of what we see in the human genome, a tremendous expansion of families, tweaking with things, and in some sense, that's what we've got to settle for our innovation.
Sexual reproduction is the third way that genetic variation arises. And basically here, the alleles have to arise usually through mutation, but the shuffling of alleles during sexual reproduction, meaning 2 parents contributing DNA, that shuffling of alleles is what's going to give you these new combinations, which give you new phenotypes. And you should know these 3 ways that the genetic variation is increased in sexual reproduction. This should be familiar from Bio 1, but if it's not, you should take a look at that again. It's in the chapter about meiosis. I'll also show you this little video.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair. greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
And just note that in asexually reproducing organisms, of which there are many, those are all not going to have this way for generation of variation. In sexually reproducing organisms, however, this is probably the most important variation generator. Gene flow very clearly leads to genetic variation. What that means, gene flow means genes are coming into or out of the population. And how does that work? You're either getting actual individuals, like these beetles, flying in or leaving the population, or maybe floating over on some sort of log. It's in some way where they're now going to breed with the population and bring new alleles in. So predominantly for variation, we're seeing new alleles coming into the population. Or you don't have to have individuals with something like pollen containing the sperm, which are gametes, or spores being distributed. If those get into a new population and they are now bringing new alleles and new complete genes into the population, and they're breeding there, then that would bring variation. Horizontal gene transfer is the last way we are going to talk about that genetic variation arises. This one is more common in bacteria, and then in this example, the hybridization of plants. I'm going to give you a little example very briefly of these different processes. We'll talk about some of them in much more detail when we get to bacteria chapter and plant and so forth. There's a bacterium. Here's its DNA. It's a circular chromosome. Let's say another bacterium has died and released its DNA to the environment because it broke open, and so you have DNA, naked DNA floating around. What can happen under the right conditions is that bacteria could pick up the DNA, and usually it would digest it, but sometimes if it's homologous enough, it could recombine it into its own genome, and now all of a sudden you have either completely new genes or at least new alleles that are now in the chromosome, and they'll get passed on every time that bacterium replicates. So that's one way to get variation. It's actually called transformation. Then we have a scenario here, I'll draw it down here. Same thing with the bacterium. Here's its chromosome, but this time we have a very enlarged virus, and it has DNA in it. And let me just mention that is accidentally picked up bacteria DNA. So your virus is supposed to have its own genetic material in there, but accidentally it picked up bacteria DNA. We'll talk about how that happens more later. But what can happen is that if the virus infects the bacterium and deposits its DNA there, the same thing can occur. Usually it's chopped up, but sometimes it could be incorporated into the chromosome, and now that bacterium has new genes or new alleles in its chromosome to pass on. Hybridization in plants, that would be when you have two separate species parents and they create a hybrid offspring that is fertile and viable, which means it can live, and fertile meaning it can reproduce. So now all of a sudden you have all these new genes combined together to make genetic variation, and especially if that hybrid can then back cross with the parent species, so then maybe they are just sort of subspecies, that'll mix all that variation around a whole lot. And finally, endosymbiosis. You might recognize that word from when we talked about the mitochondria and chloroplasts in Bio 1. And the idea there is that you have an organism and a smaller organism, and the first one engulfs the smaller one, which is not abnormal. Well, that's how they eat a lot of times. It's a single-celled organism. But in this case, instead of digesting it like would be normal, every once in a while in evolutionary history, the smaller organism, which would be a bacterium, didn't get digested, and so then it ends up becoming an organelle inside of the other organism. So it's like a cell living in a cell initially, and then it loses its own function individually. And occasionally you'll actually see some of those genes that came in with the other smaller organism being transferred into the genome of the parent cell organism and so forth, but it just brings more variation into the population as well. That's what I need you to know about the genetics behind population genetics. The next lecture that should follow this is the one about microevolution, where we actually look at how these genetic differences change or don't change over time.